Yes, good evening, everyone. Yes, so today we'll start with a new chapter, Principle of Inheritance and Variation. Yes, so to begin with the chapter, first we should know what is inheritance and what is variation. Can anyone define what is inheritance? The genes that pass from generation to generation. Very good. Any other definition? Yes. Any other definition? No? No idea? Yes. We can tell a process or phenomenon in which the character is inherited from one generation to another through factors or genes. Yes. So here, if I ask you, can you tell me what is inheritance in one word, single word? Will you be able to? I gave you the definition, a process in which the character is getting transferred from one generation to another, from the parental generation to the offspring generation. Some characters are transferred. Eh? That character is transferred with the support of factors or with the support of genes. Eh? But if I ask you in what is inheritance in a single word, how will you define? Yes, we can tell similarities. Yes, similarities. No, between the similarities between parent and offspring. Ne? Yes. Now coming to variation, variations, variations. We can tell dissimilarities. Dissimilarities. When the character is not transferred from the parent to the offspring, and when the offspring has a new character, new factor dissimilar to its parent, it is called as variations. Okay? The character, the character present in offspring not inherited from parent is termed as variation. It is called variation. Yes. So the chapter is telling us about the principle of inheritance and variation. Now we know what is inheritance and what is variations. Any doubts still here? No doubts. You know what is inheritance now? What is variation? Inheritance, in simple words, we can tell similarities. Variations, dissimilarities between the parent and the offspring. Yes, which is not transferred from one generation to another. Now, when we talk about the inheritance and variation, we are talking about factor, we are talking about genes. Yes, I give the definition where the character is getting transferred. That character is getting transferred with the support of genes, with the support of factors. That means when we study genes, what we are studying? When you study genes, what is the study called? It is called genetics. It is called the branch of biology that deals with the study of genes. No, it's called genetics. Yes. We talk about genetics. Have you ever heard who was the person to give this word called genetics? Yes, who has given the name as genetics to the study of gene? Yes, George or Mendel. Who is that new person? Hmm? Mendel? Huh? Yes, no idea? Yes. So this word genetic was given by a person named William Bateson. He was the person to give the name as genetics. Okay. When we talk about who is the father of genetics, who is the father of genetics? Father of genetics. It is Gregor Johann Mendel. Gregor Johann Mendel. Okay. Did you understand? Yes. Now, when we study genetics, when we study this chapter, principle of inheritance and variation, first and foremost, we should know about the father of genetics, Gregor Johann Mendel. Anything which you know about Gregor Johann Mendel, can you tell me, please? Yes. He very good. He did the experiment on pea plants. Any other information you? That's all. No other information. Where he was born? He was born. What study he has done? What work he has done? How many experiments he has conducted? No idea. Yes. When you are studying genetics, no, who has discovered this genetics? About him also we have to study. Yes or no? Yes. So here, for your information, when we talk about Gregor Johann Mendel, he was born in 1822. When? He was born in 1822 and in Austria. Austria. Okay. Not Australia, it is Austria. Okay. One of the country in Europe. In Europe. Okay. Nearby to Germany. Done? Yes. So he was born in 1822 in a country called Austria, which is present now in Europe. And properly, now that country is called as Czech Republic. Okay? Czech. Earlier it was called as Hungarian country. Okay? Hungarian country. Now the country name is Czech Republic. Okay? Czech Republic, which is present in Europe. So he was born in 1822. So in After a lot of difficulty, after a lot of difficulty, he passed matrix, that is SSLC. Okay? Earlier we used to call matrix. Now we call it as SSLC. No, earlier matrix. He passed matrix. He belongs to a poor agricultural family. Okay? His father was the sole bread earner for the family, and from generations, from 2 3 generations, continuously his family belonged to agriculture family. Okay? They used to work as a farmers, and they used to continue their life. Okay? So his father clearly told Mendel that he cannot continue his studies by paying more fees. So after listening to this, Gregor Johann Mendel, instead of going to higher studies, he went to the church. Okay? He went to the church by name Thomas, Saint Thomas Abbot. Okay? Famously known as Church of Brunn. Church of Brunn. So this was done in 1843. Okay? 1843. He joined as the servant, as a worker there. Okay? He was working there. After 2 3 years, he started studying, teaching there. He started teaching. Okay? In that church, in the church of Brunn, it was a small management where church is present, where educational institute is present. Okay? And other things are connected to it. So, in spite of the work, he was interested for teaching students. He went there. He started teaching also. Okay? He has taught mathematics, mathematics, geography, social science, and science also. Okay? Without the knowledge, how much knowledge he has? Only matrix pass. Is that clear? So that he started in 1843. 1843. And continuously for almost 10 years, he was working as a teacher also. He was working as a priest also. Okay, in the church until 1853. Nahi? But there, when the management members changed, okay, questions were raised against him that without degree you are teaching the students. If you want to teach the students, you should have a degree. Right? So what Mendel did with great enthusiasm, no, in 1854, he went to a university by name Vienna University. He went to Vienna University to pursue higher studies, and wrote the exam. Okay, wrote the exam in mathematics, social science, geography, science, and a few other subjects. And after giving the exams, right, in 1856, right, in 1856, he was failed. He was failed. He did not pass. He did not clear that exam. Okay. In 1856, after 3 years of hard work, he was not able to pursue that degree. Okay. He was not able to take that degree, and finally, without the degree, right, he came back. He came back to the Church of Brunn. Okay. Yes. Apart from all this scenario, no, apart from all this scene that is taking place, at one side of his life cycle, no, Mendel was interested in agriculture also. Why? Because of the generation to generation family. work. Okay, he has some knowledge related to plants, and he has the passion to cultivate the plants also, right? So here in the church of Brunn, no, so backside to the church of Brunn, the place which was empty grounded, right? In the evening time he used to cultivate plants. He used to cultivate the plants. Okay, and the favorite plant of Mendel was pea plant. Okay, it was the favorite Mendel time, that is Pisum sativum. Pisum sativum he used to cultivate. Okay, now once he returned back in 1856, eh, after the failure, he was very sad. Okay, to forget that failure, no, what he started, maximum time he started giving in the conducting of experiment, cultivating the plants. So at that phase, from 1856 1856 till 1863, almost 7 to 8 years, he has conducted experiments, he has cultivated the plants, conducted hybridization experiments, hybridization experiment, okay, on pea plant. Yes, how many experiment would he have been conducted? Any idea? Yes, any idea how many experiment he has conducted on hybridization process? No, what is hybridization? When you cross two different plants, you will get a new species, that is a hybrid. No, so hybridization by taking many characters he has conducted. No, so almost he has conducted 29,000 experiments. How many? 29,000 experiments. Okay, in this more than 10,000 experiments are conducted on Pisum sativum plant. Pisum sativum, that is pea plant, he has conducted. Did you understand till here? Yes? Huh. Shall I erase? Hmm? Yes. Now, what happened, sir? In 1863, he was ready with his experiments. Yes, he was ready with his experiments. So here, he took the experiment report, all the experiments which he has conducted. The report was made. Okay, record was taken, and he has submitted in the big university of that time, that is Vienna University. He has conducted the experiments and submitted to the Vienna University. Okay, everything he has mentioned. Okay, question arise, why you have used pea plant? Now, for conducting experiments, more than 10,000 experiments he has conducted. Question arise, why you have taken pea plant as the favorite plant? No? So he has the answer for all. Number one, it's a bisexual plant. Okay, it's a bisexual plant. Short lifespan, fast growth, economically cheap, easily affordable. So, few reasons he has given. Then finally, the report was submitted. Listen here. Now it is very crucial phase of Mendel's life. Okay, Mendel's life, crucial phase. He came back from the university without degree. Again, he continued teaching also. Again, he has conducted the experiments. One work he has got failure. Nahi? After working hard, he got failure, but he did not stop. Okay. Another side, he came, he took passion, he selected passion. Okay, and he started doing hybridization experiment, and after conducting for 7 to 8 years, he has made the report out of it, and he tried to submit the report. Okay, that report which he submitted, no, again it was rejected. All the experiment, all the work of 7 to 8 years was failed. It was fail. Failure took place for second time in Mendel's life. Okay? In Mendel's life, he faced the failure. Okay? Accordingly, he got some success. Okay? He was made abbot of that church, because of its experience. Abbot करको बोले तो father of the church. Okay? He was made, high rank was given. Okay? He was made the father of the church, and he was the in charge of that church now. Okay? A small moment of success was seen, but the real hard work which he showed in education, pursuing degree, that was also fail. Okay? That was also fail. And second, when he used the passion, he selected passion, and in that passion also he tried to gain some success. Okay? That was also again a failure. Is that clear? Now question arises, sir, so many experiments he has conducted. So many experiments, 29,000 experiments he has conducted. In that, more than 10,000 experiments conducted on pea plant. Okay? He has a very good knowledge on pea plants. He is from the agricultural background. He has conducted experiment for 6 to 8 years. Then why the result was rejected? Why it was failure? Yes, did this question come into your mind? Right? It will come, right? The reason is, right, first thing, whatever the experimental report Mendel has submitted, right, before you submit the report, before you do something, now we have social media to show that this is coming, coming soon, very soon, right? So like that, in that phase, he has not done any publicity of the experiment what he has conducted. Only few nearby to the society of Mendel where he was living, no, others does not know what Mendel actually has done, what experiment he has conducted, what results he has given, what it will be, whether it will be useful or not. Nothing is known, no information. Okay? Because of lack of information, it was not publicly spread. One. Okay? Second, if it is not publicly spread also, if it is correct, if it is through later, no, all will come into awareness if something is correct, no. But here, after the submission of the result also, the Mendel experiments were fail. The experiments were 101% correct. Whatever the experiment he has conducted, 101% it is correct, but they were rejected. The only reason is, whatever the result he has given, no, once we conduct any experiment, at the end we will give result. Nay? If I ask you what is your result in first year PUC, what you will tell? You will tell 50%, 60%, 70%. Nay? You will tell. Nay? That result what you are telling is in the mathematical form. Nay? If I tell you I am 100%, that means that 100% is mathematical form. 20% that is mathematical form. Instead of that, if I tell you my result is 3:4, 1:2, 9:3, 3:1, will you accept? Yes. The university also did not accept. Why? Because whatever the result he has given, it was in the statistical form. Statistic form. Okay. And that needs explanation. The result needs explanation. After submission, Mendel is not available to explain to all. Because of that reason, because of no explanation, okay, the statistical form result made Mendel to be disapproved. He was rejected, experiments were rejected, and finally, after this failure in 1865, ne, in 1865, he left hybridization experiments also. He left everything he left. He was working as a father of the church now, and for the improvement, for the development of the church, he continued his life. Okay? And finally, with that failure, two times in life, in 1884, he died. Okay? In 1884, finally, he died. Okay? Yes. The story will not come to an end here. Okay? Story will not end here. Why, sir? Nahin? So here, three more scientists, three more different scientists, from different countries. Okay? By name, Hugo De Vries, Carl Coren, and Tess Kermack. Okay? So three different scientists from three different countries. Yes, they were also conducting the experiment on hybridization. They were also working on genes, okay, working on factors, working on law of dominance, law of segregation, law of purity of gametes, law of independent assortment, completely related to the genes. The study was conducted by these three scientists, okay, and incidentally, no, they reach Vienna University. They came to Vienna University. Okay. Why they have come? To submit the report of their experiments conducted when? In 1900. In 19, he died in, they came to Vienna University almost 16 years later. Almost. हाँ, अभी जो experiments में हमें पढ़ती नहीं, all the experiments what he has conducted, it is in the statistical form. Okay, 1 is to 2 is to 1, 1 is to 1. Nahin? So if you ask me the result, if I tell, sir, 3 is to 4, nahin? It is difficult. Because of that, the result was rejected. Okay. Now, in 1900, after 16 years, after 16 years of Mendel death, these three scientists accidentally or incidentally, somehow, from different countries, they came and gathered here in the Vienna University to submit their experimental record. Okay. And they have also done the same experiments what Mendel has conducted. Okay. When they submitted the record, they got the information that someone earlier has done the experiment, and that record is also present in the university. So what they did, before submitting their record, they took that data from the university to cross verify whether they are correct or wrong. Okay. So that record what Mendel has submitted, when they took this record, and they started verifying the experiment conducted by Mendel and the experiment conducted by them, they came to know that many mistakes were conducted by these three scientists. Okay. By seeing the experiment of Mendel, all the mistakes were rectified, and with the proper result, with a new submission, they submitted the report. Okay. When they did this in 1900. Okay. Once they submitted the report, all the experiment were accepted by the Vienna University, and they were awarded the Nobel Prize. They were awarded the Nobel Prize for conducting the hybridization experiment. Done? Yes. But they saw the work of Mendel, and their conscience told that the real person to be rewarded is not us, it is Mendel. No? So finally, that Nobel award, what they got, no, it was named on Mendel, Okay, and the tag was given to the person called father of genetics. Okay, unfortunately Mendel does not know. Okay, even till now Mendel does not know that we are talking about him, and he is called as the father of genetics. Okay, these three individuals got the name as father of genetics, but they did not accept. They gave that name as father of genetics to Gregor Johann Mendel. Is that clear? Did you understand? This is the short story of Gregor Johann Mendel. Now, coming to the experiments, what the Mendel has conducted. Shall we proceed? Yes. So, conduct the experiment. What Mendel used to do, he used to take the characters. Characters. 7 characters he used to take. So when he take this 7 character, more 7 characters will be contrasting characters, opposite. Okay? 7 real characters, 7 contrasting character. So here we can tell that characters with 2 forms. One with the dominant and one with the recessive form. Okay? Yes. Number one, character number one, stem height. Stem height. Yes. So before going to this, we need to know some terminologies. If you know these terminologies, easily we can learn the experiments conducted by Mendel. Number one, a gene. Number two, locus. Number three, allele. Number four, homologous chromosome. Number five, dominant. Number six, recessive. Number seven, phenotype. Number eight, genotype. Number nine, homozygous condition. Number 10, heterozygous condition. Done? Many more are there, but when we conduct the experiment, one by one we'll proceed. Yes. Now, first thing, gene. Yes. If I ask you what is a gene, what you will tell? Yes. We studied inheritance. I use the word gene inside that definition. Yes. What exactly gene is doing? It is inheriting character from one generation to another. We can tell a basic unit of heredity. Gene is the basic unit of heredity. Did you understand? Yes. Number two, locus. Okay? Locus. Assume the gene is present here. The gene is present here, or the gene is present here. No, this particular position where the gene is present, it is called locus. Locus is the position. It's a place or a position where the gene is present. If one it is present, one gene is present, we will call it as gene. If a pair of gene is present, yes, we will call it as allele. Yes. Now, this gene or a pair of gene present together on the chromosome. No? So, it is called homologous chromosome. Homo means similar. So, same chromosome. Homologous chromosome. Yes. Next, coming to dominant. Dominant. Yes. What do you mean by dominant? Two genes are present together. One gene is able to express itself one gene present but unable to express itself because it is suppressed by the earlier gene. Is it clear? Example, take one glass of water, take one glass of milk. Nay, add that water to milk or milk to water. What is observed? Milk is observed. Yes, water is not present there. If you add water in the milk, nay, you are able to observe milk. You have added water. That means water is not present. It is present but unable to express. The gene which is unable to express is called recessive. The gene which is able to express called dominant. Done? Did you understand? Yes. Next, phenotype, genotype. No, phenotype, external appearance, and genotype, internal constitution, internal genetic makeup, you can tell. Okay. Yes. How to understand this, sir? Phenotype. No? Here. If I write the first character, stem height. Yes. Stem height. Stem height. What is coming into your mind? Height. Height kate ke sath icha? Tall. Nay. Against tall? Dwarf. Nay? Yes. So, this is phenotype. This is phenotype. Now, tall will have two genes. Capital T, capital T. Draft will have two genes. Nay, small t, that is genotype. Did you understand? That is genotype. Then, if, see here, if I take this capital T, small t, and from this, if I take one capital T, if I take one small t, now what I am getting? Nay? If I take this capital T, the condition is homo. Both are same. If I take this capital T, small t, it is heterozygous condition. Done? Did you understand? Yes. Now, coming to the characters what Mendel has taken. Number one, stem height. Number one, stem height. Then, number two, seed shape. Number three, seed color. Number four, pod shape. Number five, pod color. Number six, flower color. Number seven, flower position. Yes. See here, these are the seven characters, no? Seven real characters what Mendel has taken. So to these seven characters, we will have totally 14 characters, 14 contrasting character, 7 dominant, 7 recessive. See, assume seed shape. Okay. If we assume the shape of the seed, one will be round, one will be wrinkled. Yes. If I take seed, yellow, green. If I take pod, what is pod? Yes. What is pod? Don't know. We use the word pod for fruit. Okay. Yes. Pod shape. Pod shape. Yes. Inflated. Constricted. Means regular shape, irregular shape. Then pod color. Green. Yellow. Yes. Flower color. White. Violet. And flower position. Axial. Terminal. Yes, these are the seven characters to which we have 14 contrasting characters, seven dominant for conducting the experiment. Shall we proceed? When Mendel started with the experiment, he has proposed three. He has proposed three law. Okay, law of dominance, law of purity of gametes, law of independent assortment. Finish. Shall we proceed? Yes, first experiment was conducted by Mendel on pea plant, that is mono hybrid cross, or also called as one gene inheritance. One gene inheritance. Yes. So here, to begin with the experiment, Mendel is telling us it's a mono hybrid cross, mono, or it's a one gene. Ne? So what Mendel has done, he has taken one character. He has taken one character as height. Ne? So when he took this one character, so this one character will have two possibilities. It will have two possibilities. If I tell height, the first thing that comes into your mind is tall, and the second thing is draft. So what Mendel did, he took two plants, two pea plants. Okay, two pea plants, parental generation, with homozygous condition, homozygous tall pea plant. One he took. Then along with this he took one more homozygous dwarf pea plant. So two plants he took by taking one character as common. Right? Then he took the phenotype of this plant. Phenotype of this plant is tall, and phenotype of this plant is dwarf. Done? Yes. Then he took genotype. Genotype. Yes. For tall, what will be the symbol? Capital T. Capital T. For draft, what will be the symbol? Small t. Small t. Done? From this, he took the gametes. He took the gametes. One gamete he took from here. One gamete he took from here. So capital T, small t. Two gametes. Yes. Then he conducted a process called hybridization. Okay? Or cross pollination. He conducted. Okay? So when this capital T and small t was crossed together, was fused together, so what is the result we got? One capital T, one small t. Right? The result what we got is called as F1 generation. Okay? Or filial generation. Okay? And the product what we have got, this product is called as hybrid. It is hybrid. Since we have done the hybridization experiment, okay, two different plants we have taken, so therefore the result what we get is a hybrid. Did you understand? Yes. Now what you will call to this hybrid? You will call tall plant or you will call dwarf plant? Tall plant, whether it is in a homozygous condition or heterozygous condition. Yes. Now we will call it as heterozygous, heterozygous tall pea plant. Done. Did you understand till here? Everything is clear. Any doubts you have? No doubts? Yes? Huh. So, by conducting this experiment, Mendel has proposed two laws. Number one, law of dominant. Law of dominance. Okay. According to the law of dominance, when a pair of genes or a pair of allele are present together, the dominant gene will express itself and the recessive gene will be suppressed by the dominant gene. Dominant gene, recessive gene, both are present together. When the time came, the dominant gene is expressing itself. The recessive gene is present along with the dominant gene but unable to express. Okay? So with this he proved the first law of Mendel, that is law of dominance. Done? Now coming to the F2 generation. Yes, so after conducting these experiments here, he took the F1 generation. Again he conducted the same experiment. Again one more time he will get the same result. Yes. Now both the F1 generations, two times which he conducted, he took and did selfing, self pollination. Selfing was done. Done. Now if you take this, yes, capital T, capital T, capital T. Right? What is this capital T, capital T? Homozygous tall. Homozygous tall. Okay. Capital T, small t. This is heterozygous tall. Done. Capital T, small t. Again heterozygous tall. Okay. Small t, small t. Homozygous dwarf. Yes. Yes. So with this second law of Mendel was proposed, that is called as law of purity of gametes or law of segregation of gametes. What is this? You can see what we have taken two plants. One is tall, one is, we got one tall plant, homozygous condition. We got one dwarf plant, homozygous condition. Parental generation came back. Along with it, other generation also has come. So here, according to the law of purity of gametes, what Mendel is telling, when a pair of genes are present together in the F1 generation, if the blending is not happening, if the blending does not take place, only they were present together, blending did not took place, that means in the F2 generation they will segregate apart. They will segregate apart from each other. So this is what happened. In the F1 generation, if you see, we took two parental generation, one tall, one dwarf. We got the combination as heterozygous tall. It was not taken, but we have got the result. Again, when we take the heterozygous tall, in this heterozygous tall plant is also present, dwarf plant is also present. We got the result was homozygous tall, homozygous dwarf. That means they were present together in the F1 generation, they have not mixed up. Blending did not took place. Okay? and when the time came in the F2 generation, they have segregated. That means they are showing purity of gametes. If they mix up, purity is gone. They are present together, but they did not blend. They did not mix up. So the gametes were pure and they were expressed in the F2 generation. Two laws were proposed when monohybrid cross was done. Okay, one is law of dominance, second is law of purity of gametes or law of segregation of gametes. Yes. Now coming to the result part, yes, we need to take two results. One is phenotypic ratio and one more is genotypic ratio. So here, what is the phenotype we have taken? What is the phenotype? Yes, tall is to dwarf. Yes. How many tall plants are there? Three. How many? Three. Homozygous tall, heterozygous tall, heterozygous tall. Three are there. How many dwarf plants are there? Only one. So here we have got the result as 3 is to 1. This is the phenotypic ratio. Now coming to the genotypic ratio, capital T, small t, and what is the result of F1? Capital T is to capital T is to small t. Yes. How many capital T we got? Only one. How many capital T we got? Two. How many small t we got? One. Yes. What is the genotypic ratio? 1 is to 2 is to 1. Did you understand? Yes. This is the F2 generation result. Done? Yes. After conducting the experiment, we can verify the result by doing the test cross. Test cross. What is test cross? Whatever the result we get from the F1, no? F1, when we take the result, that result we have to test with recessive parent. Okay? F1 generation has to be crossed with the recessive parent. Capital T, small t, small t, small t. Now, if you cross, oh, yes. So, if you cross this, capital T, small t, small t, small t, capital T, small t, small t, small t. So here you are getting 2 is to 2. That means 1 is to 1 is the ratio. When you do the test cross, whatever the experiment, since we have taken one F1 generation, one recessive parent, we have to get back one recessive parent and one head F1 generation. Okay? So 1 is to 1 is the ratio. Did you understand? Yes, any doubts you have? It's a very easy question and scoring question. 5 marks. If the question is asked, it will be asked for 5 marks. Okay? Sometimes it will be asked for 3 marks. Majority of time it has been asked for 5 marks. ಓಪನ್ ಮಾಡ್ಕೊಂಡೆ ನೀವೇ ಎಲ್ಲದಕ್ಕೂ ಮಾಡ್ಕೊಂಡು सात को बाजू करूँ मैं बैठ खाली जरा चेक करते जरा तब क्या करते हैं चेक करो लिखा छोड़ो चेक करो खाली क्या जी लिखे करके बोलो इमरान सर बोले ना जो भी टेस्ट प्रिपेयर हो सब आती हो ना करके बोलो तुम्हें जाने टाइम को तो भेज करो और इनको तुम्हें है ना जो बच्चे नहीं है उनको ये दे डालो 10 क्वेश्चंस है ना इम्पोर्टेंट क्वेश्चंस सैटरडे के दिन ना ग्रैंड टेस्ट है तुम्हारा टेस्ट है फिजिक्स केमिस्ट्री मैथ्स बायोलॉजी कंप्यूटर साइंस हाँ तो 10 अब मैं देता हूँ तुम ना ये अब ग्रैंड टेस्ट कर वो एवरी वीक करने से छोटे छोटे करने से 10 क्वेश्चंस देना शुरू करेंगे अब तो मैं मुझे 10 क्वेश्चन वो बच्चा को दे डालो को एक कॉपी मुझे दे डालो मैं वो टाइप कर लेता हूँ क्या दियो दियो लिखेंगे दियो उनको मेन हमें वर्क लेनी मेरे को मालूम होना उसके वास्ते अब उनको एक मैसेज जाता है कि है ना इम्पोर्टेंट करा रही करके बोल को हाँ 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 तो मैं 10 क्वेश्चन सब सबका नहीं ले ले को ये सैटरडे को ग्रैंड टेस्ट कर उनको अनाउंस कर डालो जाने से पहले ग्रैंड टेस्ट था तुम ना करो तो मैसेज कॉलेज में भी स्प्रेड होती है और टेस्ट करा रही हूँ उसके वास्ते अभी एक 3-4 दिन कैच होने चाहिए वहाँ 15 की बैच पक्की करता हूँ मैं वो छोड़ता नहीं उसके वास्ते ठीक करा लेते Shall we proceed with the next one? So here, when the Mendel conducted this experiment, shall I erase? So what Mendel has done, he has taken one character. Yes, one character he has taken. That character was height. Right? He took the character as height. So two possibilities he conducted. He took the two possibilities, and in the two possibilities, tall draft. He concluded that tall is dominant. Okay, and the draft is recessive. Yes, but if any work is conducted, any experiment is conducted, so there, along with the advantages, disadvantages also will be there. Okay, negative thoughts, negative people, negative results also will be present. Right? So same thing arised when he proved the law of dominance, law of purity of gametes or segregation of gametes. One more scientist by name Karl Koren. He told that not only law of dominance is correct. The law of dominance, what you have proved, that in if two genes are present together, one gene will be dominant, another gene will be recessive. There can be one more phenomena also. There can be one more possibility also, that is called as incomplete dominance. And he conducted the experiment. Karl Koren was the person to conduct the experiment called incomplete dominance, and to prove this experiment as incomplete dominance experiment. Done? Yes. After namaz we will complete this. Okay? Yes. Our law of independent assortment that we will study when we study dielectric force. Okay. Just cross when we conduct the experiment is correct or wrong. To see that we have to do test cross. Okay. Once the experiment is going to be ratio 1:1. Okay. Dielectric 1:1 is 1:1 is 1:1 is 1 ಬೇಕು ಆದಾಗ then it is correct. I'm asking you, I will complete this topic. You complete. Okay. Yes, shall we proceed? Yes. So here, according to Carl Coran, there can be one more phenomena that is called as incomplete dominance. Now, what is this incomplete dominance? Yes, in a pair of genes or allele, if both are unable to express their dominance, when both the genes are unable to express their dominance, results in a new intermediate hybrid. Did you understand? Yes. So to prove this experiment, to prove this phenomena, Mendel took a example of snapdragon flower or dog flower, that is Antirrhinum majus species. Antirrhinum majus species, or we can also take Mirabilis jalapa, that is 4 o'clock plant. So to explain, to prove this, phenomena, he has taken this and he has conducted the experiment. He took the parental generation, yes, parental generation, two plants, two flowers he took. One is homozygous red flower, one more homozygous white flower, homozygous red and homozygous white. So phenotype of these flowers are red and white. Then coming to the genotype of this flower, that is capital R for red, and for white we will take small r, small. We can take small w also, but as printed in your textbooks, we need to take smaller, smaller. Is that clear? Yes. Now, we need to take the gametes from this. One gamete from here, one gamete from here. Yes. Then we will proceed with hybridization experiment, that is fusing. Now, cross pollination. Cross pollination is done. So we will get the result as capital R, small r. Capital R, small r. Now, what will be the result according to you? We have taken homozygous red, homozygous white. What can be this capital R and small r together? Yes, this is the F1 generation we have got, nay, and the hybrid what we have got. Yes, yes, it should be heterozygous red. Yes, it should be heterozygous red, or it should be heterozygous white. Nay, if it is a one gene inheritance, it should, one gene has to prove law of dominance, one gene has to become dominant and one gene has to become recessive. But here, since it is incomplete dominance, neither of the genes are able to express, both are unable to express their dominant, we have got the result as heterozygous pink flower. Heterozygous pink flower. So here, see, when we are taking a pair of genes and alleles, when both are unable to express, we are getting the result as intermediate. Yes, that is the intermediate. Either we have to get red color or white color, but what we have got is that we have not taken only. This proves the incomplete dominance. Done? Yes. Shall we go with the F2 generation now? Yes. Capital R, small r into capital R, small r. R, 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 R. Yes. Capital R, capital R, capital R, small r, capital R, small r, small r, small r. See, we have taken one homozygous red flower, we got back. We have taken one homozygous white flower, that also we got. This is pink color, heterozygous pink color, heterozygous pink color flower. Done. This is the F2 generation. Yes, phenotypic and genotypic ratio shall we write? Yes, phenotypic ratio. Phenotypic ratio. We have to take, no, red is to pink is to white. How many reds we have? How many red? Only one. Only one. How many pink we have? How many white we have? If we take the genotypic ratio, R, capital R, small r, small r, small r. Again, this will be 1 is to 2 is to 1. Done. So, with this result, Karl Koren has came to the conclusion that it is not law of dominance, it can be law of incomplete dominance also. There is a phenomena called incomplete dominance also. If both the genes are unable to express, this condition can arise. Is that clear? Yes. So two experiments we have conducted. Okay. Two more very important experiments. That is one more conducted by Mendel. That is a dihybrid cross, and one more experiment codominance. Right? That we will continue in the next class. Done. You can do the test cross for this also. Okay, but here which is dominant, which is recessive, we don't know. Nay, in the monohybrid cross, exactly we know that this is the recessive one. This parent will be the recessive one. But here we don't know. Nay, if we don't know which is the recessive parent, assume if I take this, okay, or if I take this unknowingly, that will become back cross. That is called back cross. Shall I erase this? Yes, see. We have got capital R, small r. Yes. Now with this, I am taking small r, small r. Capital R, small r, small r, small r. Capital R, small r, small r, small r. Nay, capital R, small r, we got 2. Small r, we got 2. 1, 1. 1 is to 1 we got. Nay. Now, if it is known, it is a recessive parent, then if we take, it is a test cross. Okay. We don't know. In the incomplete dominance, we don't know who is dominant, who is recessive. In that case, this is called as back cross. Instead of taking this, we can take this also. See, capital R, small r, 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 capital R, capital R. So R, R, capital R, small r, R, capital R, small r, R, 2, capital R, small r, 2, 1, 1, 1 is to 1. Done. Unknowingly, if you take, it is back cross. Knowingly, if you take only recessive parent, F1 generation into recessive parent, it is called test cross. Done? Yes.